Okay, and we are recording now, so this is the second session in the second unit of the course about image manipulation, so image manipulation or computer vision 2. And today we are going to be first looking at project number 2 in some detail, especially at the data related to project number 2. And then we are going to be reviewing slowly the notebook about how to process images as if they were NumPy arrays. We will define what NumPy arrays are, and we will do some basic manipulations using those NumPy arrays so that we can solve some tasks that will be very helpful for project number 2. So let me just start probably by running these cells. This that you can see in your screen is the second project on image processing. And we can start running. And I'm going to start downloading the data. Let me check in my other screen while that's loading some of the data that I already have downloaded, because that will be quicker. Okay. So slowly. This project number 2, you have a couple more weeks to submit it. Some of you have already done, as I said before, which is very helpful. First of all, let's have a look at the imports. And here you have, first of all, matplotlib that we already had for the image basics unit. That's only for visualizing the data inside of the notebook. And that's going to be very helpful because, well, our data here are no longer tables, tabular data, structured data like we saw in project number 1, but here we are using images. So making nice plots and being able to visualize these different parts of our data will be very useful. We have OS and timeit. OS is a library to handle files, to copy files, make new folders, delete things, so on and so forth, which we will use a little bit for things like file names and so on. Timeit is used to measure the time it takes to run code. And we could go really deep here, well, or a little bit deeper at least, between wall clock and processor clock and even GPU clock time, but we won't. So we will be measuring basically wall clock time, and that will be enough for our purposes. But if anyone is thinking about things like, hey, the time that it takes to run the code is a little bit difficult to define, and it depends on how many processors you have and what else the computer is doing, yes, you're right. And we can talk about it if you want. Just send me a message. But we will keep the subject as simple as possible. We also have CV2 to the OpenCV library, which will help us open and store images from the drive. NumPy is going to be very, very necessary, as we will see today. And then in the last part, especially for advanced students or for those who are a little bit more successful if they are beginners in the project, we have pandas again, because, yep, we can use pandas with images too. Specifically, we can collect information from our images, and then we can use pandas to express, to modify, to manipulate that collected information. Usually putting images inside of pandas data frame as they are is not such a good idea. We have our friend the default dict here, and we even have regular expressions, which we will use very, very seldomly. In the first cell, I have downloaded the many images, many folders, lots of data, okay? And you can see it here. Remember, we are running this notebook inside of a distributed Colab machine. So nothing of this is running in my computer now or in yours if you're running the notebook alongside me, which I actually recommend. Instead, we are each connected to a different Colab machine and running things there. So when we download something, we download it there. Okay, so here we have this data Sarah folder. Okay, Sarah is someone originally named Sarah Kench, now Sarah Brandt, because she is German and got married. And she used to be a PhD student at the Smart Forestry Group, Smart Forestry Lab at the Faculty of Agriculture. She finished in 2021, I think. And as part of her PhD, she collected lots of data and she, as part of a team, that too, and she did lots of annotations. And we are still using those annotations to see how expert annotations can help data science people process them and gain insight from the data. Basically, Sarah collected data from different sites, site 1, site 2, site 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, so 13 different sites where we collected data. Those are forest sites in the Ensuring forest near Tsuruoka. And we can open some of them, and inside of each of them, okay, that's site 11, that's site 3, we have lots and lots and lots of files. Okay, I will show you some of these files now from my computer. I can open them here. Okay, I'm opening a mosaic here. But as you can see, it takes a while. Okay, I think it's a little bit clunky because the Colab computer is not very powerful. So I'm just going to open them in GIMP in my own computer. Okay, so let me move this thing here. So now you can see folder within my computer, my local computer. I downloaded that to my local computer. And here we can just open one of the sites. Okay, same structure, lots of files. And here we want to see the structure of our data that's explained in the notebooks of the course and a little bit in the project notebook too. But basically the same thing, the first thing that we have is something called number of number of the site, S6 in this case, .jpeg, and that's the mosaic. I open it with GIMP. And after a little while, we will see one of these mosaic images, okay? This mosaic image is like a very detailed picture taken with a drone of a section of a site in the forest, okay? So you can see it here. If you have never seen a mosaic, let me tell you about them a little bit. You will see here probably these white holes hold your attention, and some of you who have keener eyes will see that here images look a little bit weird, but let's for the time focus on a different section. Okay, going down here to this section with some trees where I can see some water puddles on the floor. Okay, and here we can recognize some trees, and I can go deeper and deeper. And this has, for example, a small image artifact, and deeper. Okay, and deeper up to where things start to get pixeled out, but where I can already recognize the texture of tree leaves. Okay, so basically this is an image that shows a pretty large part of a forest, if I zoom out, but that has very good spatial resolution. So every pixel represents, I think here it's something like 1.8 centimeters. Okay, that's a lot, and that's something that only drone and similar imaging platforms allow. Before, typical resolutions for satellite data, which was what was used mostly for this type of work, of forestry work, could be 50 meters per pixel, 5 meters per pixel. If you go to something that was less than 1 meter per pixel, that was considered very, very high satellite resolution. Okay. So drones allow us to image areas of the forest that are still fairly large. Of course, satellite images larger areas, so the two image modalities are complementary. But in drones, we can still see pretty wide areas with very, very high spatial resolution. Okay, so that's what the type of imaging that we will be working with. The thing is that in order to obtain this type of images, what we do is that we fly the drone over the forest, and the drone captures many, many images as it goes flying. So what we are seeing here is not just one single image that would very likely not have such high resolution, but a composite of different images. So the drone flies a little bit with the camera pointing downwards. That's called the nadir direction, and then takes a picture. Flies a little bit more and takes another picture. Flies a little bit more and takes another picture. And flies following a flight plan. Most flight plans follow like a kind of an, how would I say it? S trajectory. So this way, and then this way, and then this way, and then this way, okay, side to side. And maybe when we have reached here, then we start going up and down also in this kind of S direction all over the side, so that each particular part of the forest is photographed at least a few times. Then we use some photogrammetry software to glue together, to paste, to join together all of those images. So we end up with something that's a composite, a mosaic that shows a large area but has very high resolution. Sometimes we mess up and we don't have very good matching, or in order to do we have some perspective deformation on something and we have to just hammer things in. And then we end up with areas that have no data, so we just let them white, or areas that have very little data and it's not very high quality, so we have to pull data from far away and we have to do this kind of weird elastic deformations, okay? That happens the most at the borders of the mosaics, of the sides, okay? Because usually what we have is that we are interested in the central part of the data collection, and then we have the areas in the extremes, in the borders, where we are not so interested and we don't care so much. So if the data there is not good, we don't care so much. Okay, this is the type of data. Nowadays, I'm not an expert in this, but I think that people at the forestry group are using also kind of spiral type of flight plans. So they begin at the center and they go doing kind of circles, but with increasing radius. So spirals, okay? So that is the type of image data that we are going to be working with. Trees in the forest, about between 1 and 2 centimeters per pixel, and showing different sites, okay? But in computer vision, and most especially in deep learning, we cannot do much if we don't have expert opinion. Data science projects need to happen in teams. Some of you here listening to me are more on the programming and computer vision side of things. So you will be more familiar with all of those coding things and so on. Some others are more on the forestry engineering side of things, and you will not be as familiar as coding, but maybe this data makes more sense for you. My main point here is that we can work together. And the more we know about the other person's field of study, the better the results will be. Forestry people, you don't have to become pro programmers, okay? You don't need to. And programming people, you will never be experts at forestry and you don't have to, okay? It's okay. But if everyone can understand a little bit of the problem that the other person has, that of the area of speciality of the other person, then it's much easier to have a common language and to bring one of these data science projects to satisfactory results. Okay. Here we are trying to create those hybrid profiles of people who know a little bit, who are still have a strong area of specialization, but can reach out to other areas. So in order to use that expert knowledge in our processing, we need to incorporate it automatically. So we asked the specialist, in this case, Sarah, to annotate the species in the site, in each of the sites. Okay, so here we have lots and lots and lots of files, and many of them are called something like S6, S0 something, okay, or S6, S13, like this, okay. Again, S6 represents the site here, site 6. The second S means species. So if I open this as a layer, S6, S13, what I get is not much, but I get a binary mask where black means there is information and white means background. Okay, so these black pixels represent the positions of trees of species 13, because this is S6, S13 in my mosaic. Okay, so here is where these trees are. This is not very helpful, so let me use transparency to show it a little bit better. So now we are seeing that the trees of species 13, and I did have a table somewhere mentioning what species that was, but I don't have it anymore. Well, I do have it, but I don't have it here at this moment, so please don't ask me what type of trees these are. But the forestry expert, Sarah, in this case, annotated that all the trees in species 13 in this particular site are here, okay? If we open another site, we may or may not have a species 13. If we have it, it will be marked in the same way, a binary mask with the black pixels indicating the presence of trees of that species and the white pixels indicating background, so not that tree species, okay? I can open another one, so another one as layer, and I'm going to open species, I don't know, species 4 now, okay? Site 6, species 4, again, let me give it a different, different transparency value. I think I did 20 before, so let's do 40 now. Yeah, okay. So, for example, here I'm seeing that these trees are from the lighter one, so species 13, and these trees are from species 4, okay? And so on and so forth. I could add all of the layers here, and I would have this map of what trees I have there and where they are, okay? That's the main information that I have. I have the RGB image that represents my mosaic, and I have all of these binary masks that represent the positions of trees, okay? Positions of trees, that's a little bit tricky because how many trees are here, for example, okay? So honestly, I don't know. It looks to me like this must be more than one tree, but I honestly do not know. Okay, so we also asked Sarah to please make a point that shows the position of the center of the canopy of every tree present. Okay, not every tree of every species, sorry, not every tree of one particular species, every tree of every species. We have that in the treetops ROI file. Okay, so we have the S something JPEG, we have the S something, S another something, which are the species files. Okay, all of those, and then we have two more things. The one that we are looking now is this treetops file. So let me open it. Okay, so this is just a bunch of points marking the center of all the canopies corresponding to different trees. Okay, let's give it yet another transparency value, 30, for example. Okay, and now I can see that there, where I was looking, I have not one, not two, but three trees. 1, 2, 3. Okay. I don't have precise separation for every tree. That would have taken more time than we had at the moment. But I know that we have some trees here and we have their treetops. Okay. And of course, every different species has its own trees. So here we have a treetop, so pretty big tree from species 13, sorry, and these are the treetops from species 4. And many, many tree tops that are from species that I have not opened the mask for. Okay. So that's the description of the data that we will be using for project number 2. I don't know if anyone has a question now about this data. Okay, as usual, if the question comes later, feel free to ask it on the forum or by email. I neglected one file. Let me open it quickly, which is the ROI file too. Okay, file, open as layers, and I can open it here. Sorry, S6 ROI, region of interest. Okay. Remember when I said before that we are only interested in what happens at the center of the mosaic? Well, we have actually made ROIs, so regions of interest for all of our sites. So this means that this part, darker now because it's in the black part of the region of interest, this is interesting. This is the part that we are interested in. And this other part that has the artifacts and so on, not so much. Okay, it's inevitable to get this kind of imaging artifacts at the border of your mosaic. So the trick is to just make a mosaic that's wider and have your region of interest in the middle. Okay, so we are, to be fully precise, going to be working with the region of interest at the center of each site. Okay, so that's it for the data description. Let me go back to the project notebook. And again, here we have the same structure, data Sara, all of the sites, and inside of every site, all of those files, S2 JPEG, the mosaic, S2 ROI, the one I have just showed you, all of the species masks. Okay, let's click on one. Binary mask showing the positions of all of those, in this case of the trees of species number 9 inside 2, okay? And then we have the treetops. Okay. Some of them also have a boxes file. We will deal with that one later in the last session of this unit. Okay. So here we have just some more description of the data in terms of files, so the folder structure. Here you can visualize the file structure if you want. So this is some, okay, what's going on here? Well, that's not working. I will fix it later. Basically, I think I'm missing an import there. I will look it up. Okay. Basically, this is something very lateral for those of you interested in how to build directory trees directly in Python. Okay. So, but I see that something is broken. Let's not dwell on it. Basically, this makes a list of the files in each directory and prints it in a pretty way. That's that's easily, let me see. I think I'm missing this import. Let's see if I can quickly fix that one. Okay, so nothing. Anyway, fix it later. So that's basically to check the structure of the files in a prettier way, but we don't need to do that at this moment. Okay, let's go with the tasks. As you have probably seen, if you have read the notebook, we have, again, a helpful code for the difficulty of things, D1, D2, D3, okay? D1, everyone should be able to solve and should be very easy for people who have previous programming experience, okay? D2 should be challenging, but even beginners should be able to do them with some work. And difficulty 3 are more challenging. Okay, so first of all, our task is to load an image using OpenCV. And I have even left some of the functions that we used in the image processing basics notebook. So you can use those or you can use some others. Okay, so I have left this read mosaic and read binary mask images here. So first, I ask you to load image S1 JPEG in folder 1. So you need to know how to either do it from scratch on your own or figure out the path to those images to call these images, okay? Task 2 is about loading binary mask. In task 1, we load one mosaic, and in task 2, we load one of these species masks and want to visualize them using matplotlib. And we want to superimpose this species mask over the mosaic to get to something like what I did before, okay? So we wanted to have something along the lines of this, okay? Inside of Python without having to use GIMP or anything similar, okay? In task 3 is where things start to get slightly more complicated, and we want to see how we can use Python to process that information that the mask gives us about the position of the trees, the pixels that correspond to every tree species. In particular, we start with something simple. We want to count the black pixels in the mask, in the mask for S1, S01.jpg that we have just loaded. Okay, we are going to do this in 3 different ways. We are going to use it with a simple for loop. You saw how to access pixels using double indexes for our images, because images are matrices of pixels and matrices are just lists of lists. So images are by transitive property lists of pixels. And we want to count how many of those are black. Okay, that means how many of those has the value of 0, because this is a binary mask, so it only has one, well, grayscale mask in particular, but we use it as a binary mask. It has only one value for every pixel. If it was a RGB image like we load here, it would have 3, the red, green, and blue. This one is grayscale, so it only has 1. And we want to count how many pixels are black, so how many have 0 value. Easy peasy, double for the 2 dimensions of the images, of the image, of the binary mask, and then count those that are 0, okay? Double for with an if inside. Problem is that, well, that might not be the most efficient. So we also try different things. Here you can use a map. This is slightly tricky, okay? So if you're a beginner and you're getting really bogged down here, just skip it, okay? The map part. But for experts, for people who have some experience, this is not very difficult to do, and I encourage you to do it. And then we will use it. We will use NumPy to do the same thing, to count the black pixels in the image, okay? What is NumPy? I'm getting right to that in a second, okay? So first, let's say serious task 1 and 2 are very easy. Task 3, a little bit more difficult. Count the black pixels in one of these grayscale masks, pixels with value 0, using a 4, using a map, if you're up to it, and definitely for everyone as NumPy arrays. Measure the time for the 3 things and compare, and comment, okay? I have very little use for things that only have code that do not have any comments on them. The point is that you understand what you're doing and think about how to use it, okay? Someone just, I don't know, coding the thing and not thinking about it, or even worse, asking ChatGPT or Claude or whatever and not thinking about it is not much use to them or to me. So please think about the results, comment on them, okay? Then we move to the large-scale data manipulation, where we will create a label image, okay? I will introduce this very briefly and then move on to the NumPy thing. But basically what we have here is that, as we saw before, we have our data divided in lots of images, okay? So here I'm showing just 1 and 2 species masks, okay? But I have more. I also have the ROI, and if I open them as layers, for this particular site, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 16, 17, 18, 19, 20 different species here. There are, I think, 44 possible species, which are not always present in all sites, okay? So species go from, I would say, 0, 1 to 4, 4. Check that one, it might be 4, 7, okay? So check with your data. You can do that automatically, or you can just open the folders and see. And then we want to manage all of that separated data in a way that's a little bit more convenient. So what we will build is a label image. We will build one single image that gathers together all of those information. And what that image will do, we'll talk about it more next week, but it will collect. So for example, if this is species, which one is this one? That's species 4. I want to create a label image, an image that has the number 4 in all of those pixels, okay? In all of the pixels corresponding to species number 4. 0 or nothing in all of the pixels corresponding to no 3 species. The path here, okay, the ones outside of the ROI, there I want a 0. And wherever I have a 3 species, I want the code for that species. For here, there I have species number 13. So all of these pixels should end up having a number 13 in them, okay? We could just open all of the images at the same time, do a really long chain of ifs there. If I'm in this image, then do a double for and gather all of this image and put the correct number in the correct pixel in the result image. We could do it that way. That would be very, very slow, okay? So instead, we will use NumPy vectors to solve this task, to NumPy arrays, okay? So I have introduced task 1, 2, and 3 with some detail, and task 4 with less detail, okay? Task 4, I hope that everyone can complete, but we will speak about it in more detail next week, okay? Then we have task 5 about the data sanity check, which is a direct consequence of what is done in task number 4 to detect multiple annotations and then do in task 6 census type statistics, okay? For beginners, passing the subject, you should be able to complete at least task number 3, okay? And then if you can go task 4, 5, and maybe 6, you can get to the 100-point mark. Okay. Task number 3 is usually where people stumble and can be a little bit frustrated. Sorry, not 3, task number 4. Okay, so 1 to 3 should be fairly easy, and then 4 should be where you finish passing the project, and then 5, 6 where you earn more marks. Okay, and people stumble at task number 4, a little bit at task number 3, and then more at task number 4. But let's give the tools not to stumble so much and to solve that properly. And those tools are in, let's see if I can get it here somewhere. Not this one, this one. So in this, sorry, in the course notebooks, I'm going to use the one about NumPy for image manipulation. Okay. You have it in the general description of the course, in the updates post, and I'm going to run it here too. Okay. Again, I'm downloading the data, and it's exactly the same data that I downloaded a second ago, and well, not a second, a while ago, and I spent lots of time talking about. Okay. Here I'm listing the files again and running some. So it takes a little while to download things and so on. I'm just using the same images that I left for you in project number 2. So the images to read mosaics, so RGB images and binary masks, grayscale masks. And here, for example, I'm reading one of them. I'm defining a path first and calling a function read mosaic to read one of those from my data. Okay, my downloads have already almost finished, 98, 99, done. Okay, good, extracting. We have to allow for the fact that this is actually quite heavy data. Okay, so here we have some more details on the files in one of the folders, the functions that I mentioned before, and here we have called one of the functions and visualized it with Matplotlib. Okay, this might be useful for task number 1, and we can do things. I'm not doing it now, but here, for example, we take advantage of the fact that this is an image, and we can rotate it if we want. We can modify the matrix of pixels, and then we get all of the proper results. Okay. As we saw last time, we can also show sub-image by selecting some part. You will also need to do that for project number 2. And then we finally get to NumPy vectors, okay? NumPy arrays. So let me very quickly just open some code here. And let me, as you can see here, I have defined some path, okay? Actually, I think I have it, what I need a little bit, yeah, here. I can copy it directly from here, okay? So we can open and show one of those masks if you want, okay? I have called readVM to show one binary mask. I have read it from the disk of the Colab machine. So here somewhere in S2 and S201. So I'm using this path library to create this path for the images, and then I'm reading them using matplotlib. I could also have done something like path equals, so the data folder is something like data Sarah. This has to be a string. Sorry, all of it has to be a string. Data Sarah, and then S2, and then S2 S01.jpg. That, sorry, extra string here, that would have worked too, okay? If I had used this path to read this, would have worked too. Why did I use this os.path.join command? Well, because this one is system dependent. This one works on the Colab machines because they are Linux machines. In Windows, I think this should be a slash probably instead of a bar, okay? So in order to make this system independent, I just call it with this, and I tell this library to join the different parts of the path with whatever my machine is using, and then the library takes care of that, okay? So a bit of, again, of a more advanced thing, but basically I get the same result. I read and show my binary mask. And what takes time here is the showing, so I'm going to get rid of it. As I said before, I can just access some of my pixels with double indexings for my lists of lists, okay? Can print one of them. Okay, so perhaps unsurprisingly, that 1, 2, 3 is white because most of the things here are background, so that's going to be white. As I said before, if I want to, for example, count the black pixels here, I can go for a double 4. I need mask 1 shape, not weird things, okay? And I can print that one. That's going to give me the columns and rows in my matrix, in my image, okay? So if I go for I in the range of mask shape of 0, so the first component, in this case 9945, and then I go also for J in, this is range, not rank, of the second component, then I can print all of the pixels in my mask, okay? And now I'm going to get a really long list of numbers. That's going to, it's difficult to see, but it's growing, okay? So many of them are white, and if I wait long enough, I will also get to see some zeros there. Okay. So actually it's only showing me the last 500 lines because Colab got tired of showing values. I think I actually have some zeros in here. Well, difficult to show them. But basically here, I'm traversing all of my pixels one by one, okay? And I can do that, and I'm still doing it, okay? So I'm just printing them and not doing much with them, but the list is growing, and it takes a while. I'm going to stop it right there, okay? So you can see lots of 255s, lots of pixels that are white, and occasionally you also find some that are black. If you wanted to count the number of black pixels here, you could do it like this for task number 3, I think, okay? But as the experience with running this loop that I have just made already tells you, this is going to take a while. So we really would rather not be doing this. how can we get code that achieves our goal, in this case of counting the black pixels, and is faster? I'm going to delete this one. We don't need it. Well, we will use NumPy vectors, NumPy arrays. Okay. NumPy is actually not an image processing library. It's a library for processing this type of array data in parallel. Nowadays, our computers, most of the computers that we use have more than one processor, and Python is a programming language that heavily optimized to use that computing power, okay? To run things in parallel, to have different calculations that affect different parts of the data at the same time. In particular, with this loop that I was making, we are just traversing one by one all of the pixels, and just for your project, you will just need to check if each of those pixels is 0 or not. So a pretty simple calculation at every pixel, and we can parallelize that. We can make this run and check different pixels at the same time, okay? In the third unit of the course, we will do lots of calculations with PyTorch, which is a GPU parallelization library. And the PyTorch is a library that uses GPU parallelization. So you have lots of functions that run in the GPU, which is another place where you can do lots of simple calculations in parallel. Here, we want to use CPU. We want to parallelize at the level of our computer's processors. And NumPy very helpfully does it for us. If we define things in a way that this NumPy library can understand in NumPy array format, then it can do calculations for us very quickly. So let's define, first of all, a simple NumPy array, okay? We can define it here, for example, from a list of lists, okay? So, and I can probably even print A itself. Yeah, okay. Have defined a NumPy array as a list of lists, and now I have this. And this is essentially the same format that our images have, which are lists of pixels in their case, okay? And if it was a grayscale image, it would look very much like this with probably a lot more pixels, okay? Arrays have this shape function that we have been using also for our images, and data type, for example. In this case, my numbers are being stored in integer 64 format, okay? As a difference from Python lists, the elements in an array always need to be the same type, okay? Because NumPy wants to be able to process things very quickly. Here you have another example of how to build arrays from lists. Have a look at that slowly. Here we can show the dimensions of one of my images using this NumPy syntax, okay? We access one pixel, all of that works like we had been doing, okay? But now we are basically using NumPy, and we can use this D type. And for example, my image is the mosaic, so when I call the D type here, I get that the pixel is, one of the pixels is here, and the pixel type is array itself, because in every pixel I have a list of 3 values, R, G, and B, and that's stored as an array of arrays. And then each pixel inside of this, each of the value there is an uint, so an integer that's unsigned, that doesn't have plus and minus, and has 8 bytes, okay? As we saw before, here we can also load and show binary masks. We will load a few of them, okay, and we will show something about them. Here you have lots of lines where we load and show different binary masks, and you might want to look into that again for task number 2 in project 2, okay? PyTorch also allows us, sorry, not PyTorch, Matplotlib allows us to superimpose things with this alpha parameter thing that I was doing with the transparency in GIMP. In Matplotlib, you can do it with this alpha. And you have also something that you can use this for, again, in project number 2. Let's wait until it shows those 3 masks, but basically we are loading from site number 2 the masks for species 1, 2, and 3, and showing them here superimposed with different levels of transparency, 4, 6, and 8. Okay, here we have those 3 masks. As you can see, there are like 3 different levels of gray, one that's very dark, one that's in the middle, and one that's almost white. Okay, and we can show a sub-image. We can define 3 sub-masks by always taking exactly the same limits on the 3 masks that we have read, and we show those 3 masks using Matplotlib and the same level of transparency, and we will end up with a sub part of that, okay? The one that's around here, okay? Defined by the limits. Here we show a close-up of the 3 masks that we will see, that we were seeing. Okay, and we are now reaching the NumPy part of the whole thing. NumPy has very helpfully many pre-built functions that help us do things. For example, we can count the number of black pixels in an image with NumPy using this syntax here, okay? And let me just run it so that you can see how long it takes. It's done, okay? When you run the loop version, you will see that it does take a while. NumPy version is very, very quick, okay? Basically, we have defined the black pixels in mask number 1 as black NP1 and counted them with NumPy sum, and then here we have this syntax, mask1 equals 0. Okay. We also used the timeit function to define that. To count the time of that. Okay. Okay. So how did this line work? Okay, what did we do here? So first we asked NumPy to sum, to count, to sum, to do the addition of something. And what NumPy added up was this mask1 equals 0. Okay. So basically what we did here is that we first consider one of our binary masks and called this mask1 equals 0. This returns a Boolean mask of the same size of this original mask with the same number of rows and columns, and in each pixel position it returns the true or false, the Boolean value of this condition. Okay, so NumPy in parallel processes all of the mask1 image and checks at every pixel whether or not that pixel holds this condition, whether or not that pixel is 0. For those that are 0, it returns a true value. And for those that are not 0, returns a false value. Okay, so with this very simple call, not all of it, only this part, we are building a Boolean mask of the same size of the image full of true or false values. Okay, you can see it here, for example. I've made a small array with all of these values, and now I'm printing my array equals 0. Okay, and I get true in the positions that, yes, equal 0. Okay, I could do this with equals, I don't know, 128. Okay, and then I would get true only here and here. Okay. So that's what that part does. And then once I have this mask, Boolean mask of the same size, then I sum, I add it up. Okay. This might be a little bit counterintuitive because I'm basically adding up Boolean values. It's a little bit of a trick because basically when I look at a Boolean value, true or false, as if it was an integer, Python very helpfully translates true to 1 and false to 0. So if I add up a whole array full of Boolean values, I count the number of truths. Because the truths were the ones that held the condition that I'm interested in. In this case, the truths were the black pixels. Then I'm counting the number of black pixels. Okay. So by using this parallel programming to make this big mask of the same size of the image with true or false values, and then adding up all of the true values, well, adding all of them, but true count as 1 and false count as 0, then I can compute the number of black pixels very, very efficiently. Okay? That's one way in which NumPy allows me to process images and to get some information from them very, very quickly. Okay. And I can sophisticate my condition. For example, I don't have to restrict my condition to just one image. I can use more than one image in my condition. So if I do np sum of mask1 equals mask2, what I get first is a Boolean mask, Boolean image of the same size as mask1 and mask2. And if they are not the same size, I will get an error. So they have to be the same size. But luckily for us in project 2, everything is the same size inside of the same site. Okay. So this is going to go over all of the pixels and check what pixels have the same value in mask 1 and mask 2. Okay, so pixel by pixel, are these two positions the same corresponding position, the same value or not? If they are, I will get a true, and then I will count it here. And if they are not, I will not get a true. So this gives me the number of pixels where mask 1 and mask 2 coincide. Okay, and then I can keep sophisticating this if I want. Okay. I'm going to give you now 10 minutes to just go to start project number 2, okay? And see if you can get to this part about, I think opening the images should be fine. You don't have to do the visualization now, but try to get to the for loop part and to the NumPy manipulation, okay? Start thinking about it. Let's do 10 minutes and then we will give a few minutes for some questions and maybe for the conclusion, okay? I'm going to pause the recording now. Please remind me to start it again later. So 10 minutes, have a look at this. Sorry, I don't know if there is any question at this moment before the 10 minutes. Okay. Sorry, actually, I realized that I paused the screen sharing and not the recording. So now I am pausing the recording. Okay. So we are recording again for this final part of the session. So if you're watching this from the YouTube video, then that's been a very short pause, but I do encourage you to press pause yourself and take a few minutes to look into those things and to try to solve those tasks now. And for those of you who have been working at home at this moment, I don't know if you have any question. It was a short pause, so you probably did not have time to finish everything, but I hope it gave you some time to at least think about how to start. Okay. I did not have time to provide complete feedback for the person who submitted the project. I did see that the error that I got during the running of the Colab notebook, they also got. So please do not worry about that initial error. I will fix it, and I will upload the fixed version. So do not worry about that directory, that folder structure display thing. I think there we are missing one package that used to be there and is no longer there. So do not worry too much about it. Then, as is usually the case, this person had a pretty good grasp on the initial tasks that are the ones that you can directly find in the notebook that we have been seeing, and then, well, stopped at the slightly more difficult tasks. So try to do those. And especially, I think it would be very helpful if you all had tackled or tried to tackle task number 4 before next week. My recommendation is that you do task 1 and 2, which will be very quick. Task 3 is going to take a little bit more, but you have had some time to think about it and then ask me questions, and that should be really easy to do. Well, not really, but doable within this week. And I recommend that you start thinking about task 4, that one about building the label image. Okay, I will talk about that one in detail next week. I will also mention a little bit more about this mask image processing in the context of not drone images, but document images, because we are now, we have fresh out from the oven the image processing course, document image processing course, and I will want to show you a little bit using that data too, so that you can see that the techniques that we are learning here can be used in different applied projects. Okay. And as I think there are no questions at this moment, okay. So I'm going back to um to the notebook that we were seeing, to just to a little bit of next week regarding this NumPy processing and how that relates to the remaining tasks in the project. Not task 1 to 3, we have already seen those. Task 4 is a direct consequence of what we have already seen, but it bears a little bit more commenting. And then task 5 and 6, okay? Those you can do using slightly more complex conditions like the ones that we have seen. We already saw this one, that I can count the number of pixels that are the same in two masks, but I can do, I don't know, number of pixels that are black at the same time. Okay, so not the pixels that are mask in one, that are black in one mask, or black in the other, or the addition of two, but the pixels that are black at the same time in the two masks, or the pixels that are black in one mask and white in the other. Okay, by using this type of composite conditions, I can process information in any way that I want. And this is going to be very quick because I'm using NumPy to do it. Okay, so this would be the both black, not many. And this is important for task number 4. Okay, where we try to detect overlapping annotation. That should not happen. Every pixel must, in theory, belong to either one class or the background. But because this is human annotation, there are always small errors, small inconsistencies, and we have to find a way to deal with them. So this is something that we will deal with in task 5. Then we can just time some of the counting that we are doing. For example, going back to the basics, we are counting the numbers of pixels that are black in mask 2 and 3, counting them, okay? Getting very, very similar time readings for them. Why are they slightly different? Why are they not the same if they are two masks with the same size and the same shape? Well, because this time, as I said before, is clock time, and maybe the system in the Colab computer was a little bit more busy the first time around than the second, and we had these very small differences, okay? As I said before, we can get into how to measure time sometime later. Okay, and I can keep using NumPy. For example, I can make a copy of my image using the copy function. That's also a quick way of doing these copies. And then I can do different things. For example, I can create a mask image, a color mask image, and I am masking the pixels that are 0 in mask 1 as red. So I'm giving them 25500, and as this is, sorry, not, this is RGB in, yes, in Matplotlib, so this is the red mask, okay? So the pixels that are 0 in mask 1, I'm going to paint them red. The second one, RGB, are going to be green, and those are the ones that are black in mask 2. And the third one, RGB blue, are the pixels that are black in mask 3. I show them, and then I have a visualization of the pixels that are black that belong to each three species. These are three species, 1, 2, and 3. Okay, an easy way to visualize this. And to do it, I'm again using this idea of paint all of the pixels that are black in mask 1. This is a Boolean mask, and I'm using it to modify this my image C. And I'm putting this new value, this 25500 value there, okay? To build the label image in task 4, we are going to use some of these principles. We are not going to end up doing something like this, like modifying this, but we are going to be using the pixels that are black in one mask or in another, and we are going to modify them, okay? And I don't have time anymore now, but we can detect overlap. So this is going to be very helpful for task 5. So take some time to read this. We might get back to this next week or the other, maybe. Here we have a function to check double annotations, which might come in handy for task 5, but you need to understand and adapt it to your situation. And even we can show some census type statistics, which is what we do in task number 6. Okay. In this, we try to count the number of pixels in each mask and combine them and, for example, put them inside of a pandas data frame. Okay. So basically, I hope that in this session you have seen that manipulating and modifying images with NumPy is quick. It's not difficult. It has its own syntax, which you have explained in detail here in this part, okay? Sorry, in this part here of images and NumPy vectors, and I said before, this has changed since last week, so please do have another look if you already checked this before. That tasks 1, 2, and 3 in the project are pretty simple, especially 1 and 2. And that for task number 4, you should understand this manipulation as NumPy vectors. We will talk about it a little bit later, a little bit more in detail next week. Task number 5 and 6, you have quite a lot of material that might interest you in this same notebook. Okay, so that's it for today. I don't know if there is any question at this moment. Okay, keep them coming via, as I said before, the web class forum or the Slack channel or by email, okay? We are going to finish the class now, and thanks everyone for listening.